Hello everyone, this is Dr. Todd. I hope you have had a very nice week 12 in Genes and Disease. Now a number of you have been asking questions specifically about lesson 12-4, nail patella syndrome. This assignment seems to be quite difficult for many of you, and it has everything to do with gene linkage. So I'm just going to do a really brief tutorial to try to help you understand this concept. And I'll give you an example problem. Okay, we are going to be talking here about two linked genes. These two linked genes are A and B, and they are both dominant. So when a child would inherit the dominant A allele, they will have the disease. When they inherit the dominant B allele, they will inherit the disease. So here we have our male, here we have our female. And we can see their genotypes. Remember, there are two chromosomes. Let's just call this chromosome, let's call it chromosome 2. So on chromosome 2, we have these genotypes in our male and our female. So in terms of the parental gametes, our male can provide an A, B, or an dominant A, dominant B. And our female can provide an A, B, these are both recessives, or a dominant A and a recessive B. Next, let's look at the recombinant types that our male can provide. Well, you can get crossing over during meiosis. And in meiosis, when this happens, you would have a dominant A, B, which you can see right here, or you would have a dominant A and a recessive B. In our female, we would have the crossing over, and we would have AB and AB. Notice that the parental and the recombinant types are the same in the female. Now we want to remember that these two genes are 20 map units apart. This means that they cannot independently assort. Independent assortment is out of the question. This is because these two genes are on the same chromosome, and actually they're not that far apart. They're only 20 map units apart. What this means is that the recombinant in our male, these recombinants will be produced 20% of the time. 10% will be of this genotype and 10% will be of this genotype. In our parent, then we will have a possibility of 80% getting the parental, because this must add up to 100%. So we're going to have 40% of this genotype and 40% of this genotype. The same holds true for our female. So we're going to have 10% here, 10% here, for a total of 20%, and we're going to have 40% here, 40% of the gametes of the dominant A and recessive B, for a total of 80%. I hope this makes sense. Now, let's ask ourselves a question. What is the possibility that a child from this male and female will not have either diseases? In order for a child not to have either disease, they would have to have this genotype. I hope this makes sense. 2 recessive A's and 2 recessive B's. In order for this genotype to arise, so no disease of either type, no diseases in the child, in order for this to happen, our male would have to provide a recessive A and a recessive B, and our female would need to provide a recessive A and a recessive B. So all we need to figure out is how often will this gamete be produced in the male? Well, if we look over here and we look for the recessive A and the recessive B, we're going to see that there is absolutely no chance that this can come from the male. This cannot happen. If we look at our female, we will see that the recessive A and recessive B can occur in 50% of the time, 50% of the time. So basically, if we asked ourselves, what is the chance that a child of these two parents is going to have no disease, there is no opportunity for this to occur. No opportunity for this to occur. We could ask ourselves then any other question about any other genotype, and all we would be doing is basically looking at the chance that the gametes, the specific gametes, would be produced. For example, wonder if we were looking for the possibility that we would get the genotype A, B from our male and dominant A, little b from our female, for the final genotype as the AA, little b, meaning that the child will have disease A but will not have disease B. So I'll put a little check mark here and an X here. In order for this to happen, we just need to figure out how often is the male going to give us the dominant A and the recessive B. In this case, this is going to happen here, and it's going to happen here. So there's a 50% chance for our male to provide this genotype type of gamete. In our female, the chance for this to happen, a dominant A and a recessive B, is going to be 40%. And if we look down here, we have the recombinant type of 10%. So there is a 50% chance. So the chance that we are going to get this genotype is going to be 50 times 50, which gives us 25% chance. So in this case, there will be a 25% chance that we will get this genotype. That ends the short tutorial, and I hope that it was helpful. You should be able to follow this video, and it should help you complete the assignment 12-4, or Nail-Patella syndrome. Just write out the parental and the recombinant gametes, figure out the chances that you're going to get these gametes in both the male and in the female, and then ask yourself, how often are you going to get the correct gametes to get a certain genotype as AABB, so the recessive, homologous recessive, or we get the homologous at the dominant A and the homologous at the recessive B. Thank you.